Well, thank you very much, ladies and gentlemen, and it's a great pleasure to be back in Australia and Sydney. I usually try to come here once a year if I can, but it's not always that I can find the money or get paid for coming. This time I managed to put together a package that meant that I shouldn't be too much out of pocket. So I'd like to start off really with the talk isn't entirely about science, business, or even the future, but it's really different aspects of antibody pharmaceuticals that I've been involved with over the years, and perhaps I can, give you some idea of the ebb and flow of science, business, and its implications. So I'm going to talk about antibody pharmaceuticals, and what I thought I would first of all do is to remind the non-immunologists of the features that antibodies have that make them excellent therapeutics. They are in fact nature's own therapeutics. I'm showing here an IgG molecule, a general Y-shaped molecule. In this case you can see the 4-chain structure as 2 heavy chains which are marked in red and 2 light chains which are marked in white. At one end of the molecule here, the V region, they bind to antigen. And by these, of course, are raised to infectious agents, viruses or bacteria, and by binding to the pathogen, that may be sufficient to block its action. So you can think about a toxin. The simple act of binding with high affinity with an antibody may be sufficient to neutralize a toxin. In other cases, particularly when one comes to use antibodies against human tissues, that act of binding, in particular the bivalent binding of having the 2 ends capable of binding to different cell receptors on the surface of a cell, can lead to triggering the receptors, or indeed to loss of the receptor from the cell surface, or it can drive apoptosis. So once again, the simple action of monovalent or indeed bivalent binding can be enough for its therapeutic purpose. The other feature antibodies have is they stay in the serum for a long time, 14 to 28 days. And they achieve this in 2 ways. You might say a passive way and an active way. In the passive way, because they're large, above 150 kilodaltons, they escape filtration in the kidneys. However, that isn't enough if you're a protein in blood. What normally happens is the cells, the endothelial cells, and particularly in the liver, are constantly being subjected to pinocytosis. So essentially there are cells there that are constantly sipping the serum. It's not receptor mediated, just simply sipping the serum, taking it into the cell and destroying any protein contained in there. Now, clearly if you're making a therapeutic, you don't want to have those proteins destroyed, and there are specialized mechanisms that have developed for recycling the antibody back to the surface of the cell and pushing it out again, but nevertheless destroying the entity to which that antibody may be bound. So essentially, by virtue of being bound to a recycling receptor, it is another contributor to the decent half-life of antibodies. And to give you an idea, the sort of contribution of the size and the recycling receptor, perhaps if I tell you that if we made an antibody fragment, for example, the size of the FAB, well, usually let's say the 2 variable domains here, about 25,000, and we put that into a mouse, essentially it would have a half-life of less than half an hour. It would come through the kidneys. If, on the other hand, we took that mouse, we took a knockout mouse which lacked the recycling receptor, so it's unable to do the pinocytosis, and we took a normal full antibody, which you'd be expecting a 14 to 28 day half-life, that will have a half-life of about a day. So that's telling you the two factors are multiplying. Essentially, you have one factor taking from half an hour to a day, which is the size, and another factor, which is presumed about 14 to 20-fold, which is the recycling component. So both of these things contribute to that very long serum half-life. And in fact, that ability to stay in serum for a long period of time at a decent concentration is one of the things that has made antibodies very attractive for therapy. Essentially, you can maintain a blockade of an antigen or a receptor for very long periods of time, which are not possible with small molecules, which essentially, many of them will have half-lives in the hour or two period. Therefore, you get an oscillating curve, and it's very difficult to maintain the blood concentration, certainly for those periods. So we have binding antigen, we have the long half-life, and we have killing. So this other end of the antibody here, the so-called Fc end of the antibody, acts as a flag to the immune system. So anything to which this antibody is bound, it brings in killing molecules and killing cells. It brings in complement C1q, it brings in neutrophils, macrophages, and NK cells. And the exact sets of receptors it engages in the subclass of those killing cells depends on whether these are of the IgG1, 2, or 3, or 4 isotype. To give you a flavor of what's going on here, if you look, generally neutrophils, macrophages, and NK cells are the main killing cells, and the sort of distribution of the specialized receptors on the surface of these cells, and I've marked here the relevant distributions of Fc gamma R1, 2, and 3. So particularly for NK cells, the Fc gamma R3 is the most important receptor, and this is probably one of the major killing mechanisms, certainly for killing human tumor cells. But if you look at the whole plethora of activities that one particular kind of antibody, an IgG1, can engage, we can see there are these 3 cell types, and essentially it can give it a burst of superoxides and peroxides. This is really equivalent to kind of the molecular equivalent of a toilet bleach being trucked at the bug. You've got a straightforward eating process, phagocytosis, and this actually involves the bug being put into an acid bath and then ripped apart by molecular grinders and machines in the cell, the proteasome. And we have a series of toxic molecules such as TNF, and we have ADCC, which is the antibody-dependent cellular cytotoxicity mechanism mediated by NK cells, where you get release of perforins and granzymes close to the membrane of a pathogen, or for that matter, a tumor cell, which take it out that way. So one antibody has these multiple therapeutic activities by binding, hanging around in the serum, and triggering these. Now, in some cases, it's very difficult to know what the key mechanism was, and in fact, pharmaceutical companies, in many cases, I know IDEC spent a lot of time trying to find out. And I remember talking to the CEO of IDEC on one occasion about it, and he said he doesn't care anymore. He just, basically, it just works. And that's all he says, that they've wasted so much money, he just doesn't want to think about it. So, what I've shown you there is the behavior of antibodies in their natural state, but their application to dealing with essentially becoming pharmaceuticals was only made possible by the development of various technologies. In the first case, when antibody response is made, it's a very complex thing, lots of different antibodies, and the first advance was the creation of mouse monoclonal antibodies by Milstein and colleagues. However, it was very difficult to create human antibodies in the same way as those mouse monoclonals, and therefore, with the advent of genetic engineering, it became possible to think of ways around this, and in particular, antibodies were developed which essentially take the antibody binding activity from the V regions and of the mouse antibody and put that onto the constant regions, or the Fc portion of a human antibody. So in this case, this antibody is 1/3 mouse, 2/3 human. An approach I came up with are called humanized antibodies, and these, we just transplanted the antigen-binding tips, the little loops here at the end, directly into a human antibody. A little bit more complicated, but actually these antibodies you could regard as synthetic human antibodies, because these regions vary between one human antibody and another, and all one's doing is taking the binding activity from a mouse and grafting it in there. And genetically we do this by creating a synthetic gene, so it's essentially a human variable region with just the loop spliced in by synthetic DNA. And again, I won't go into the technologies involved, but there were at least 2 major technologies now which make it possible to make human therapeutic antibodies. One of them using large antibody repertoires of human antibody genes and combining them to create very big libraries and selecting those using phage display. And another approach essentially has been to genetically engineer mice with human antibody genes. So for this talk, I'm not going to say any more about it than just to say, look, there's a whole plethora of technologies that were developed and were essential for this technology to go forward. Each of those technologies has contributed to a series of therapeutic antibodies. This shows the timeline from 1994 to 2010, and the biggest class actually are the humanized antibodies still. The chimeric being more immunogenic have generally fallen out of favor now, although there's some very big sellers in this area here. And we're now seeing a wave of human antibodies. The first one was from the phage display, and one of the last ones was phage display, but a lot of the others were made from genetically engineered mice. The fact is, this technology works. There are multiple ways of making antibodies that are good enough. There's no evidence, in fact, that the human antibodies, in any case, are better than the humanized antibodies. Essentially, there's a range of technologies that can be used. So this just gives you a flavor of the kind of effects one can see with these antibodies which have been human or humanized. Now this first thing is quite interesting. This is actually the very first treatment of a patient, and this was a patient in Cambridge in a collaboration with Herman Waldman. We made a humanized antibody against rat lymphocytes. That's against both the T and B cells. And this was the patient with non-Hodgkin's lymphoma, and this is the extent of the tumor. When we gave it for a total of 30 days, we were able to destroy, as far as we could see, all of that tumor. Now this one here, this line here, this red line, is actually the 10-day line. And that actually illustrates the importance of humanizing antibodies. Because when you take the rat antibody directly, you provoke an immune response in the human that blocks the therapy if you're using a simple rat monoclonal or mouse monoclonal antibody. So effectively, it allows you to continue that therapy for much longer than you would be able to otherwise, to the extent that you can maintain a long-term treatment without getting the immunogenic response. Now, there are a series of antibodies that have been made to treat cancer. I've listed there some of the cancers, the breast and bowel cancer in particular, and also, of course, the various types of leukemias. Another area where antibodies have been very useful, and I have to say those have largely involved antibodies which bind and kill. In the case of immune disorders, the antibodies, some of them are just blockers, but others are binders and blockers. So, for example, this shows a patient with plaque psoriasis being treated with another antibody, adalimumab, which is otherwise known as Humira. And you can see the dramatic effect in this case of dealing with this inflammatory process. Now, Humira is generally exerting most of its action, as far as we can tell, by simply blocking the action of TNF. There's no killing involved. Yet there'll be other antibodies. For example, an antibody, the use of this antibody here, alemtuzumab, has been used recently for treatment of multiple sclerosis by essentially killing the inflammatory cells that would migrate and cause a multiple sclerosis. And that antibody is an antibody that kills. So there are situations where you can either, by blocking an inflammatory effector or by killing the cells involved, you can achieve therapeutic benefit. What this shows you is that the sales, now these antibodies have actually done incredibly well in becoming blockbusters. By that, people mean more than $1 billion per year. And in fact, if we look at the total top 10 therapeutic products, and this is in 2010, because this is the last year I could get the information free without having to pay someone for going into a database. So this here is the number of billions of dollars, US dollars, and in 2010, Lipitor then was number one. And actually I grouped all the insulins together just to give you an idea of what the proprietary insulins contribute. Advair, which is a small molecule, a beta-2 antagonist, it's agonist, a beta-2 agonist, is up here. This is used for treating asthma. But then all the ones in black are antibodies. So in fact, you can see here in the top 10 pharmaceutical products in 2010, 6 of those were antibodies. And in fact, the situation's changed a bit. Subsequently, Humira is now replaced Lipitor. Lipitor, one reason it's replaced it is Lipitor has come off patent, so therefore it goes off a patent cliff. But actually, Humira is also up, I think, now at about $9 billion per year. So the actual sales have gone up. So you can see that suddenly these antibodies, which actually really, I think you may have noticed on the slide, didn't come in until '94, have suddenly stormed the top end of the pharmaceutical market. Now, there's one reason for that, and that's because they cost a lot. So therefore, you get very good sales figures. But actually, the costings are quite interesting. I mean, you can see a whole swathe of costings here of sort of, the 30,000, sort of 50,000 range. But then you will see others coming in at much higher costings per year. But there's also one here of $1,000 per year. Now, one of the arguments made about antibodies is they're expensive drugs. Well, there's nothing intrinsically expensive about them. The cost of goods is higher than with chemicals. But you may ask, how can one drug sell for $1,000 per year and others be $600,000 per year? That's not cost of goods. It is, of course, profit and the market. And so if you look, for example, at that first one, denosumab, being used for treatment of osteoporosis, this is against the rank ligand. That antibody has to compete with bisphosphonates, in particular soluble bisphosphonate, which is given once every 6 months or once a year. And therefore that antibody has been, because the amount of rank ligand is small, that antibody is injected into patients and it lasts about 6 months in a patient. So there's one injection every 6 months. And in fact it goes through 6 half-lives, but there's still enough in the blood to be able to neutralize rank ligand. Now, the price for soluble bisphosphonate is about $700. Since actually the antibodies, the denosumab, it may be slightly better than a bisphosphonate, but actually not so much better that any provider is going to pay $600,000 a year. The fact is, so far the evidence is it's good, and it therefore has to compete with chemicals. It has to compete with an established market of a soluble bisphosphonate, and that's why they're only getting $1,000 per year, but hopefully a big market. We move up to a more typical range. We're dealing with these sort of metastatic colorectal cancers, but actually I want to just distinguish between these two. Trastuzumab is Herceptin, that's against the HER2 antigen on breast cancer. And that is currently about $54,000 per year. But, and I'll come to this later, that this antibody, there are certain patients who do not respond for one reason or another. And in that particular case, by putting an immunoconjugate of something extremely toxic on that antibody, in this case an embtansin conjugate, this antibody can now be sold at $94,000 per year. So actually that is a product. I mean, it doesn't cost them $40,000 to put a conjugate on a dose of antibody. But this, I suppose, pays for the trials, and it pays for the edge you have, those people who actually need it. We have the most outrageous price here, which is $600,000 per year for eculizumab, which is against this rare disease of paroxysmal nocturnal hemoglobinuria, where you start to destroy your red cells. And in fact, there is, since there is no other solution, then essentially you take it or you die, as far as I understand. So therefore, that's why you can charge those figures. And that actually underpins some of the pricing and the commercial success of these antibodies. And obviously what one would hope is, in light of competition between antibodies, that these prices in the end will come down. One of the other strange things about this whole area was that these are now pharmaceuticals, but actually most of the development, in fact virtually all of the early stage development, was not done by pharmaceutical companies. So all the technologies, mouse monoclonal antibodies, chimeric antibodies, humanized antibodies, and human antibodies, all of those technologies were developed either in biotech or particularly in academic laboratories. In addition, with that technology, even the interesting targets, let's say TNF. TNF is one of the most wonderful targets for antibodies. The antibodies, it actually has a dramatic effect on rheumatoid arthritis. Patients end up walking out of wheelchairs and being able to move about and no longer in pain. It's quite dramatic. And they're very successful. There are several anti-TNF antibodies. They sell about $20 billion per year combined. So it is the most successful target of antibodies so far. Now that work emerged from actually work of an Australian, Mark Feldman, who was from WEHI, working in the Kennedy Institute in London, along with Tiny Maney, who was an Indian clinician. And they worked with Centocor, a biotechnology company, to do a clinical trial in rheumatoid arthritis. They'd identified TNF might be a good target, and they did the trial with an antibody that Centocor had made for another purpose, septic shock. So I won't go through these other targets, but in particular, if we think about some of the big ones, Herceptin and Avastin against HER2 and VEGF, that early work was really done through Genentech, which was then a biotechnology company. EGFR, another big antibody, was developed by ImClone Systems. That was the Martha Stewart company with all those dodgy share transactions. But they came out a really good antibody. So how did pharmaceutical companies become involved? Well, when they saw the kind of prices you could get, they bought them all. And so basically, we have Roche buying Genentech, we have UBS, which is a Belgian company, UCB buying Celtec, we have AstraZeneca bought two, it bought Cambridge Antibody Technology and MedImmune, all of these antibody companies. Abbott bought BASF Pharma, which had worked with Cambridge Antibody to make the antibody Humira. So actually, Abbott bought BASF Pharma and AstraZeneca bought Cambridge Antibody. So the two people who made some contribution to that antibody were bought out. GSK bought Human Genome Sciences, which had developed Benlysta and a number of other antibodies. The various the transgenic mouse technology on making human antibodies from mice was developed by Abgenix and Medarix, and both of those were bought out. Essentially, so Amgen and Bristol Myers Squibb would have a platform for making antibodies. And Eli Lilly, which had taken no interest in antibodies to that time, bought ImClone and the anti-EGFRs. And the value they placed on these acquisitions was very much on different elements thought important by different companies at different times, but essentially the value elements were the technology platform, the lead product, or in fact in some cases just the fact that large pharma hadn't even got any team of biologists to do anything, and they could therefore buy an active team who could go forward. And so the values are often a composite of that. It's often very difficult to actually work out why a company bought another biotech company at an early stage. Okay, so I've described where we are and how we got there, and the question is what next? And I think what next is, I'm going to divide into two bits, targets and technology. Okay, so let's just explain one interesting target which is currently being developed, which is, and has shown good data in clinical trials, which is the use of antibodies to clear cholesterol. Now, when I first heard this, I thought they must be mad. How could you pump in enough antibody to take the cholesterol out? It doesn't work like that. It's much more cunning. It essentially shows the principle, which is that in the cells that clear cholesterol in the liver, what you've got is the LDL receptor, and it binds to LDL, which is this part of a pie in solution here. And this is the LDL is the bad cholesterol, and so what happens is these receptors, the LDL receptor, takes it into the cell and destroys it, so that's cleared. But so it doesn't suck all the cholesterol out of your body, there is a mechanism whereby these cells are also producing a molecule called PSCK9, which is this light-colored molecule. This comes out, and this molecule competes with these same LDL receptors. And so when it competes with the LDL receptors, essentially it blocks them for the LDL protein. And an antibody was developed which binds to PSCK9, thereby blocking this and allowing this process to run full tilt without essentially to remove the LDL receptor. And this antibody actually looks very good. It seems able to bring levels of cholesterol down below the level that statins would have. And I assume, again, the advantage would be in the long term that you may be able to have long-term dosing rather than having to take a pill every day. The area that's getting the pharmaceutical companies most excited, though, I'm quite sure there'll be developments of this, is the whole issue of the immune checkpoint inhibitors. So it's actually very interesting. The question is, for a long time, immunologists have been saying that actually we've got immune cells. For example, if we have a tumor, we've got immune cells on board that would actually kill that tumor, but they're switched off. And so the question is, how could you turn those cells on? Because if you could turn them on, they would clear your tumors. The argument is this is a natural process happening all the time. Tumors are rising all the time, and you're clearing them by immune mechanisms. And so a requirement for a T cell to be activated is essentially you have to have engagement of the MHC with T cell receptor, but you've also got that activation requires accessory molecules. And in fact, in particular, it requires the interaction of a molecule on the antigen-presenting cell to interact with CD28 on the T cell. In this case, this T cell will be activated. At the same time, you have an inhibitory interaction with CTLA4. So I've marked this red and this green to indicate this one tends to shut down the T cell. The CTLA4 interaction with the dendritic cell or the antigen-presenting cell shuts it down, whereas this activates it. So what's been discovered is if you make antibodies that block the binding, the anti-CTLA4 antibodies, they block this binding, and therefore the T cells, which may have been suppressed, are activated, and therefore T cells become active, that the T cells hanging around your body that actually weren't doing anything, were in a suppressed state, are activated and go and start to attack your tissues. Now that doesn't sound like a good idea, but actually if there's a lot of those which actually were made to the tumor, then you might be able to get, and if the tumor's pumping out lots of antigen that found its way into these antigen-presenting cells, then you may have some advantage. It's a dangerous strategy, it's been used, and actually it does have serious side effects, but it's clearly doing something. And for melanoma, where you clearly have some kind of level of suppression that's taken place on the T cells, it's starting to have an effect on melanoma. Now, a related area is the actual action of the tumor cells themselves. So there's a black tumor cell, here we have the T cell again, here we have the TCR, recognizing the MHC on the tumor cell. But in addition, there are receptors, programmed death receptor, which is actually on the T cell, and there is also a ligand on the tumor cell. And what happens is when these two things bind to each other, so if these are normally present, these bind, and then you will not get T cell activation in the presence of the tumor. So what tumors do is they upregulate the amount of PD-1 ligand on the surface of the cell, and this therefore means that T cells which may be activated come along and they're turned off again. So other antibodies have been developed to block this interaction here, and therefore they have been used to, can also be used to activate the, to allow T cells which might have been activated and then deactivated when they encounter the tumor by virtue of the PD-1 ligand. In fact, trials have been run with the two together, which sounds like about the most dangerous thing you could do. But actually, in melanoma, they're looking good, but with serious side effects. But nevertheless, there have been patients who've had long remissions from melanoma by use of this strategy. And there are lots of ways you could imagine developing this further, in particular, and I'll come to it when we discuss bispecific antibodies, but ways in which you could tune this process up to make the immunology just depend on your target cell rather than just general activation throughout the body. So that gives you a flavor of the interesting targets, and I'm sure there are many more which are being explored. But the other thing, I'm essentially a technologist, so I thought I'd tell you a bit about technology. And start off by perhaps just pointing out the disadvantages between antibodies and classical chemical drugs. So, for example, I've marked in green the advantages of an antibody over a chemical, which is marked in red. And vice versa, advantages of chemical over an antibody on the lower half of the slide. And to go through that, antibodies have advantages over chemicals because chemicals are quite small, they have relatively few interactions with the target, and therefore it's quite difficult to block a target, difficult to make a very high affinity small molecule that will go into a target. Antibodies have a huge surface, and therefore they can get very tight binding affinity, but over a wide surface. That same huge binding surface can allow them to have exquisite specificity. So chemicals particularly tend to have a hydrophobic dimension, and they drop in all sorts of hydrophobic cavities in lots of other proteins, giving you a toxicity which you may only pick up late in clinical trials, which is an issue for the big chemical companies. As I've said earlier, antibodies have a long serum half-life. Chemicals come through the kidney quite quickly unless they bind a bit to serum albumin. If they bind too much, that's no good because they mop up all the chemical. They recruit effector functions, which I've described, and the other virtue of antibodies is because they're big, they can block the interaction of a ligand with its receptor. So if we think about TNF is a big protein. It reacts with a protein receptor on a cell surface. There are no convenient cavities to drop a chemical in that would actually block that interaction. So antibodies are great big things. They can get in there and block it. So actually antibodies are very useful for blocking protein-protein interactions. But they have some problems. They have difficulty because they're big, getting into pockets. They don't easily get outside the blood because they're big and don't go through the membrane. They don't get into cells very easily unless you try to add on specific mechanisms. You can't take them as a pill, so they can't put them through the oral route. And in fact, you can't chemically synthesize them, which you can with small molecules. So antibodies have to be fermented, and this adds huge costs to the production, particularly early on, because you've got a biological product, you have to have authenticated cell banks, and the cost of making enough antibody to do a toxicity trial will be several $1 million. Just to be able to show it actually doesn't kill anybody or it doesn't actually cause them any damage, let alone the pharmacological activity, where you'll be up to a few $100 million. So chemicals are actually much cheaper. from that point of view. So those are the advantages and disadvantages of antibodies. So what I thought one could do is, using that as a framework, we can say, well, let's look at the approaches we can take. We could either try to take advantage of the advantages of antibodies. Could we make them better in their strong characteristics? Could we try to overcome the disadvantages? So I've marked in dark green those things where we can do better. Perhaps we can do better, or have done better. Well, in principle, let's suppose you wanted to make antibodies with much longer half-life, so you don't have to inject them every couple of weeks to a month. Maybe you could make them last a year in the serum. Now that's a very interesting proposition, because if you can make antibodies last a year, that means you can just go to your doctor once, every anniversary, go around, get your shot of antibody, and have done with all these pills. It's going to have a huge advantage in compliance. In particular, old folk who may be a little bit confused as to what they're taking and the rest of it. So actually, I think that's potentially a great advantage if we can push out the time where, in fact, once a year injection you'll tolerate, once every 2 weeks you won't. Now, how to do it? Well, one way is to improve the capture by the recycling receptor, and in fact people have done this, so there are now alleged to be antibodies made by MedImmune which have a half-life 2 to 3 times longer than a classical antibody. Now, if you imagine you've got a half-life of 2 to 3 months, then you can start to imagine if you put in excessive antibody, you may still have enough at the end of a year to be able to, you've gone through a few half-lives, you may still have enough to be able to do its physiological action. So that's being done, and I don't know how far it will go, but we're certainly at the stage of probably 2 to 3 months half-life of an antibody. Okay, so there's another way in which antibodies get taken out. That is, if they bind to a target on the surface of a cell, the cell actually can take them in and chew them up. So one way to deal with that is to take advantage of the pH change that happens when you go into a cell. So by putting on histidine residues, which will become positively charged at acid pH, what you can arrange is when the antibody goes into the cell, dragged in by the ligand that's going to get dragged in by the target, which is going to get chewed up, you can dissociate it from that target by having an antibody where the binding of the antibody to the target is dissociated by the high pH. So it protonates and they spring apart. So the antibody can then get recycled and taken back out again, and that has been developed by a Japanese group to increase the half-life of those antibodies in the serum that would otherwise be cleared by binding to cell receptors. Again, we're talking about doubling, I think, the half-life of that class of antibodies. Okay, so the other possibility is you could try to make the antibodies more potent so they could more effectively engage. I told you there are all these killing activities built into antibodies, so they're recruiting all these cells. So what about improving that? Well, again, it turns out that by you can modulate by making changes in the antibody, either by making protein engineered changes or by changing the glycosylation, creating afucosyl antibodies, that they will bind much more strongly to Fc gamma R3, which is the receptor on the NK cells, which is involved in quite a lot of killing. Now, in fact, some of those antibodies are something like 1,000-fold more potent than a normal native antibody in an in vitro experiment. The problem is that it's not, when you go into animal models, it doesn't follow through. And the reason for that is it looks as though the limiting factor, for example, in clearing a tumor, is the number of NK cells in the tumor. Quite clearly, even if the antibody is more potent, if you don't have enough NK cells to do the job in the tumor, then it's not going to make any difference. And therefore, ideally, if you could find a way of boosting the number of NK cells in the tumor, I'm quite sure that will happen, you could actually take advantage of that extra potency. You could have the toxic chemical. This is what I've described. So you could actually get direct cell killing. So I've said in some cases the antibodies, by binding to the cell surface, they can take the ligand in, they can drive apoptosis. But another possibility is when the antibody goes in, it carries in a toxin, and that toxin kills the cell. And that's what Genentech did with Herceptin. They added on a toxin, and that definitely increases its effectiveness. So in that case, you're not relying on other cells like NK cells, macrophages, monocytes, whatever, around the place to do any killing. If it binds the cell and is taken in, even if it doesn't do apoptosis, it will kill that cell by just being taken in and release of the toxin. Okay, so another possibility is is an interest in building antibodies to two targets. So you've got your standard, what I call vanilla-flavored antibodies. What about a tutti frutti antibody with extra bits in? Now, it turns out the marketing people love it. The marketing people are the keenest on this approach because they say, look, we've got an antibody and it's the same as some other companies. How do we differentiate that? We've got to show we've got extra power, extra zap, like toothpaste. It's got the signal, got the thing in the stripes or whatever. So the marketing guys love it, but actually there's good clinical reasons for doing it as well in some specific cases. So, for example, if you could build two activities into an antibody, this might be very useful for treatment of particularly psoriatic arthritis and rheumatoid arthritis. So it's been shown that anti-TNFs and anti-IL-17A antibodies individually they've got proven efficacy in treating rheumatoid arthritis and certainly the IL-17A and I think in psoriatic arthritis. And recently it's been discovered that those patients, the non-responders, and those patients who in the end start to fail treatment on the anti-TNFs, start to develop a higher anti-inflammatory activity by virtue of producing IL-17A. So you've got these Th17 cells which produce the IL-17 and they're going up in patients who are not responding well. And in that case, there's a lot of logic in putting the two together. So in that way, you would block the one inflammatory mediator and another, and you would avoid the disease escaping from the pressure of those two molecules together. Another possibility would be to put on other effector functions. So suppose you could put on, suppose you could start to think, well, okay, antibodies recruit NK cells. Maybe, why don't we recruit T cells? That'd be quite fun. You might be able to activate them, and you could then bring in a whole new arm. Now, the danger is, if you had an antibody, it could activate T cells, and it could recruit T cells. In addition, it can recruit NK cells. It could just bring the two of them together, and you'd have a kind of gigantic explosion as the two different arms of the immune system attack each other. So you'd have to neutralize so for example, if you wanted this function, if you wanted to be able an extra killing function through T cells, what you'd have to do is to put a lesion in the recruitment of the antibody effector mechanism. So it could do one or the other, but still could be interesting in some cases. Now, how to do that? There have been different approaches taken. One approach has been to produce a little domain that you stick on the end of an antibody. It's about 60 residues long. It's a company I consult for called Covagen. It's in Zurich. And that actually they've made an anti-TNF, anti-IL-17A antibody, very nicely behaved, going into clinical trials now. So we'll see how well that works. There's another antibody which is, other company called F-Star. I also consult for them, and what they're doing is they're creating binding sites by making changes in this bit of the antibody which doesn't seem to be used for very much. So you can use that, and therefore you can build another activity at this end. That's more complicated, but it also works. So there's different technologies for building extra activities into the antibody, which allows the marketing guys and the clinicians a differentiation. The only people who are not happy about it are the development people who've got to take this through clinical development, because it's much more complicated to take two specificities through and meet all those requirements. So we ended up then, now we look at our antibody versus chemicals. We've just built in all these 3 extra activities. So the antibodies are winning now. You can see we've got all the green and now we've got some blue. And The question is now, can we take out these reds? Can we actually overcome some of the disadvantages? And I said at the beginning, we will not be able to entirely, but let me just suggest how we started on this. So one idea is maybe we could shrink the scaffold. An antibody is a huge molecule. The end here is a beta sheet scaffold with plug-in loops that bind to the antigen. It's these loops that do the work. The rest of the antibody, so these do the binding activity. So actually all the rest of this is just a scaffold to stick those loops on. So maybe we could just get 6 loops and put them together on nothing, hold them together on something else. Well, then we thought that's a bit complicated, but maybe how many loops do we actually need? And experimental data suggested that probably only need 2. So quite often in antibodies, we've got all these loops here, but only 2 make major binding interactions. So let's just go with those 2. Or let's just make a molecule that has 2 loops. It should be able to do most of the things antibody binding would do. So this was the idea, and we discovered that there was a Dutch group which had made taken a peptide with 3 cysteines and added to it tris-bromomethylbenzene. And this is quite mild chemistry. Let's say for chemists, it's mild. You know, you have lots of organic solvents in there as well as water. But what they did was to do this, and you end up with this little molecule will effectively take those 3 cysteines and zip the whole thing up to create 2 loops. And we thought, well, maybe we could use this for making antibody mimics. But how would we get ones with the right activity? And what we did here was to use the phage display technology, which I haven't explained, but I mentioned briefly. It's a technology we use for making human antibodies from large repertoires of antibody genes. And the idea is that in this particular case, we took a peptide, we fused it to the end of filamentous bacteriophage genetically, so it's fused here in the genome. And then we add this chemical to it, and it cross-links it on the surface of phage. And this phage, which has now got this constrained peptide on here, we can capture those phages which have got the right activity by binding them to the target protein. In fact, what we do is we pass them down a column of the target protein, and it fishes out the ones we need. Now, that approach was proven in the case of antibodies. It works very well. You can make libraries of billions of different possibilities, and therefore you should be able to find the thing you want. The problem in this case is when we added this chemical to our phages with these cysteines on a peptide, essentially is it cross-linked all the phages and they were totally inactive. So it meant we had to tweak conditions, and quite a lot of about 2 years' worth of tweaking by Christian Heinus to get the conditions such that we only killed off 90% of the phage. But if we start from 10 billion library size, we've still got 1 billion left, which means enough leads to select something useful from. What we found is we could get these bicyclic peptides that got some good properties. We found that they could have high binding activity and binding specificity, and I've just shown one here. I don't want to go in great detail, but this is one called PK15. It has a loop size of 6 by 6. It binds to human kallikrein with about 3 nanomolar binding affinity, same to monkey kallikrein, but actually has a very poor binding activity to rat plasma kallikrein. It has required it doesn't bind any of these related human proteins. So it's pretty specific, but now let's take the case we want to develop this as a therapeutic to hit human kallikrein. You need to do at least 2 animal species if you're going to do a development of such an antibody, and therefore we can use monkey, I guess, but we'd also like to use rat. So we really wanted this molecule. We wanted a kallikrein inhibitor that would bind to both human, monkey, and rat. So how do we achieve that? We decided the way to achieve that would be to play with the size and the loop size of the molecule. And interesting enough, if we drop the loop size, what we now found was it had become smaller, much more like a small chemical molecule. And we found out we start to bind to the rat kallikrein. But in addition, we're getting an unwanted activity because we're now starting to sit into the binding site of one of these human proteases that we actually don't want to touch. And so we played around with the 3x3 loop, and in the end we managed to find a 5x5 loop that actually had the right activity. So a good activity for these 3 different animal species, but actually no binding to the other ones. And that shows how you can tune the affinity and the specificity by, in this case, playing with the loop size of these molecules. Christian Heines solved the structure of one of these antibodies in a collaboration, one of these bicyclic peptides, and this shows that this is one against human plasminogen activator, and this is actually fitting very nicely as a little flat entity into a pocket in the surface of UPA. We could see there was a multitude. This shows the bicyclic peptide. There's a multitude of interactions, hydrogen bonds, ionic bonds, with this molecule fitting in there. It's obviously highly specific binding in there. We also wondered about the properties of these molecules. One of the problems with putting peptides in or using them at all in medicine is they get chewed up. We found out that the ones we'd selected against kallikrein didn't get chewed up, not with kallikrein, and we tested them for ability to be chewed up with chymotrypsin or trypsin or just serum. And what this shows is that the antibodies, the peptides, I keep saying antibodies, the peptides were, once we'd constrained them with this trisbromethylbenzene, they were much more resistant to proteolysis than a simple linear peptide that we derivatized without the cross-link, but we derivatized with iodoacetamide. So we have about a 100-fold difference in rate of proteolysis, and in fact what we found is in serum, again, these things did not get readily proteolized. So that's promising. It looks like you put things in serum and they don't get chewed up. However, there is another problem, that these entities, you put them in serum and they're small and they quickly come out. So this shows doses in rat plasma, and so we've got a half-life of less than 30 minutes. So how do we deal with that? What you can do, these are chemicals, you can add on other entities, so we put on palmitic acid. This binds to serum albumin. The binding of serum albumin holds it in the serum. So this actually shows now we've taken those, we've injected it subcutaneously, and we're getting actually an increase to about 8 hours, and then in fact there's a half-life of about 8 hours the other way. So these things now have a much better behavior than a simple peptide. They stay alive for that amount of time in the serum, and they're not, they're held out of the kidney by virtue of binding to the serum albumin. So that may mean we can get something of a characteristic somewhere between antibodies and small molecules. Okay, so this summarizes then the different, I call pharmaceutical classes, but I thought I'd put bicycles on the end here to show where we are with them. So in green are those things we've shown already. In white are those things which we haven't shown, but have some reasonable confidence that we can show at some point in the future. We've actually put 2 of these bicyclic peptides together and shown we can attack 2 targets. We think we'll be able to direct cell killing, but we haven't shown it yet. We think we'll be able to recruit T cells, not shown it yet. We've shown a serum half-life that's somewhere between the 2. I think it'll be difficult to get it out to the very long half-life of antibodies, but you never know. The other thing is we haven't yet recruited antibody effector functions, but I don't see any reason why that shouldn't be possible. So I think in the end we will be able to fill in quite a lot of this, and these will have, we do know compared to antibodies we can get these into pockets. I showed you a picture of one of those, and we also know that we can get it outside the blood. So they're small enough they do actually start to perfuse tissues. And one of the interests we have is to use these to get to tumours, particularly for carrying an particularly for carrying a conjugate, that you'd have difficulty getting to with an antibody fusion. We also know we can chemically synthesize them, which actually makes them much cheaper, and to that extent you could have more shots on goal. So in a way, the likely advantages of these, because obviously we're not going to get all this sorted out, so we need to find, well, not in the near future, so we need to identify what clinical conditions would such molecules be suitable for. And what I've listed here is the likely advantages, the disadvantages, and therefore the possible uses. So we can chemically synthesize them. This would make it very easy to make them, even for a clinician. Just a peptide group could make them easily. One advantage of these small molecules is that they're much easier to put into slow-release formulations. Proteins are quite difficult to build into slow-release, and we think there could be a lot of interest. These should be much easier because the structure is held together by covalent forces, not non-covalent forces. You can't denature these. You could boil them and they'd still be active when they've cooled down, because essentially you're not denaturing them. They're locked together with that ring in the middle. I mentioned already they get outside the blood. The disadvantage is going to be the serum half-life, and there'll be fewer effector mechanisms, however we work it. So the uses would then be topicals. That would be skin or lung. That we can get in easily. That doesn't rely very much on serum half-life. We don't have to worry about that. You can imagine them, the area I particularly like is using them as drug conjugates, so essentially using them to direct a drug to a particular class of cells we want to finish off or regulate. You can imagine using them as a short-term antagonist. So, for example, there are various immunological flares, like in hereditary angioedema, where you just need a treatment for half an hour or so, and that might be sufficient. It will be flushed out after that time. And you could use it, again, as agonists. So you could start a biological process, trigger a receptor, and you might not want to keep triggering that receptor because you might get downregulation of the receptor. So actually, as agonists, they could also be useful. But I think for my money, I'd be focusing on killing cells, essentially by using the methods that are now people are starting to develop in antibodies. So that's the last slide, and I thought I would just sort of summarize by saying what you can see is that the antibody field emerged as a result of various engineering, in particular making monoclonal antibodies and then finding ways of humanizing them to be able to overcome the immune response. You can see engineering brings on a lot more possibilities. There's lots of other things that can be done with antibodies. So I actually don't think they're going to disappear in the near future. I think we've got them here for a bit, and I think they're just going to become increasingly part of a rather expensive series of medical treatments.